Okay, at the end of last class we had gone over 8.2 and then I assigned you to read 8.3, which is all about DNA replication. So today what I'm going to do is I'm going to talk to you a little bit more about DNA replication and how it works. Another super short section, and it deals 100% with what you learned from 8.2. Remember, the main thing from 8.2 was DNA base pairing. A pairs with T, C pairs with G. So as long as you remember that, we should be okay for DNA replication. Okay, DNA replication, the big idea here, the big picture is that DNA needs to copy itself. In order for us to make all of the new cells in our bodies, we need to somehow replicate the DNA. That's all it means. Replication, replicate, copy. Okay, when does replication occur? We have to go back 5, 6, 7, when we talked about mitosis and meiosis. It happens in that interface, in your cell cycle. You have G1, S, G2. We're going to focus right now on S, the synthesis, or the part where DNA gets copied. Okay? This happens once every rotation of the cell cycle. Okay? The whole point of DNA replication is to make identical strands of DNA. So how does this work? DNA, you have the double-stranded, and we talked about this. 2 strands. Now, it says a single strand of DNA serves as a template. So one of these 2 strands can serve as a template. Think of a template like an outline, something you trace, something you look at. If I'm an artist and I'm going to draw a picture, I'm probably going to look at something. That would be my template before I draw it out. So how does DNA replication work? The first part to DNA replication is you have to unzip the DNA. So that's kind of the first step. And it says enzymes unzip the double helix. So your double helix is all wound up like this. That does not make for an easy replication. I could not come in here and start copying the DNA. It wouldn't work that way. So what has to happen is an enzyme needs to pull it apart. So you'll notice I left this bottom part kind of undone. So we'd imagine that the enzyme started unzipping right here. And you can see they're no longer twisted together. We still have the double strand, just not that twisted strand. Now, simple, not 100% easy way is, one of these is my template. And what happens is we have all these new nucleotides just kind of floating around, and they're going to come in and pair up. So same rules. T and A go together, C and G go together, and they match themselves up. And you'll notice right here, look, we just made new strands of DNA. The template is the black. That's the old strand. That's the original. And the new strand is the pink strand. But put together, we have 2 strands of DNA. Let's look at this a little closer. It says DNA polymerase bond the nucleotides together to form the double helix. So after the double helix has been, it's kind of zipped together up here, and now it's unwinding. And we said we had an enzyme that unzips it. We now see there's a whole other part. You have DNA polymerase, and that's important to remember. DNA polymerase is specific for DNA. What that does is as all these bonds come in, and again, this combination that I am creating is just made up, but it's all the pairs. I'm always keeping them together. Okay? Well, what keeps them together? What's going to hold those in place? That's what this DNA polymerase does. So your DNA polymerase actually kind of goes up the strands and it starts binding these together. It starts creating that covalent bond on the outside. So it's creating a bond. So you have one enzyme that unzips your DNA, and you have the polymerase that goes and puts everything kind of back together. Then, when you're done, you have two new strands. Strand 1, strand 2. And you'll see, each strand is made up of one new part and one old part. So DNA is called semi-conservative because it conserves. Here's your old, so the pink is the old strand, and the green is your new. Same thing on this side. New strand, old strand. Semi-conservative. So DNA replication really does try and use what you already have. But the goal is to get those two new strands, and your body just keeps doing this. This is how you make all of the cells in your body. What this slide is showing you, so A, B, C, and D, it's showing you just another way to look at DNA replication. So in A, that would be the site where the enzyme starts. And it shows you that it doesn't have to start at just one spot. It doesn't have to be the beginning of the strand. It doesn't have to be the end. It can be the middle. It could start up here. It could start here. It could start down here. It can have many starting points. That's why DNA can replicate so quickly. And that's B then builds. So the enzyme has unzipped it, so you kind of see that bubble loop. And that loop is where the DNA is unwound. Okay, so that's the unzipping. C is just kind of showing you now it's elongating, so it's getting bigger so you can have that area you need to copy itself. And D is showing you, ta-da, we have our two new strands back to the original form. Again, because it's so quick, it needs to be able to start at many points. Think of it like Highway 20. Is there only one entrance to Highway 20? No. There's lots of different entrances to Highway 20. If there was only one entrance, that would take people forever to get on and off the highway. They need that quickness. Now, one of you last class asked me a question about, well, how do we not have more mutations? How come we don't know if we have mutations? Well, a lot of times your body fixes the mutations before you even know they happen. And it says DNA polymerase can fix and correct your errors. If I said, all right, you guys, you have a 10-page essay due next class, go. You could type on whatever you wanted, but 10 pages minimum. Go. Would you have typos? Yeah, we all would. Would you click spell check before you turned it in? Of course. Spell check is our DNA polymerase. Spell check is trying to find those mistakes. Is spell check perfect? No. Neither is DNA polymerase. That's why some mutations still occur. Think back to last chapter. You can find those mutations if you had a karyotype done, where you could look at all of the different chromosomes in your body to find the mistakes. Otherwise, it's usually not seen until you reproduce, until you have kids that might show a sign of a mutation. Otherwise, we're all fine. It's okay to have a mutation. You can live on. But that's kind of the, going back to the essay, is it's not perfect because I could write in my essay 2. I may have been referring to the number 2, and maybe I accidentally typed out TOO. Would spell check that? It would check it, but it would see that it was right because I spelled 2 right. I just used it in the wrong format. So there would be a mutation or a mistake in my paper. Would the teacher dock me an entire letter grade for that one mistake? I hope not. And that's the same way your replication process occurs. Can you carry on even though you have one mutation? Absolutely. And we'll talk more about that in section 4. The last part of today's notes, I added this in, and this is, again, you can find this same worksheet on the 8.3 notes section. But it gives you an overall picture of what is happening in DNA replication. So if we were to look at this picture, and we go through step by step, what is step number 1? The process. What are your steps? Well, the first step, and you can see it right here, unzip. Okay, you have to unzip the DNA. Well, that helps me identify the structures. If you look at number 1, I'm unzipping the DNA. All right, it's DNA. Okay, 2. What is happening in 2? Well, you can see these little pieces starting to come in. Those little pieces are your nucleotides. So what is step 2? Nucleotides pair up. Okay, let's look at 3. Now 3 right here structure-wise, it's this big kind of dark shaded blob. We have the same dark shaded area over here. What is that pointing to? Well, what's the piece that connects everything together? Just noticed you guys weren't seeing the board, so hopefully that will help. Okay, this third piece, well, that's the polymerase. And it's very important that you put DNA polymerase because there will come a time when we talk about a different type of polymerase. So you want to make sure you identify that. Okay, so what's the third step? The DNA polymerase forms new bonds. What's our end result? New DNA. Now, because I talked about this in the lecture notes, I want to make sure you see it here. The end result, we have 2 identical strands of DNA. That's our end result. 2 identical strands of DNA. All right, that finishes up our notes for section 3. We'll go over this in class. If you have any questions about anything, please come in and ask me for help.